Thanks, Jeremy. Thanks, everybody. Hello. My name is Mark Harvey from the Western Australian Museum, and I'll be presenting today on a group of pseudoscorpions on behalf of my two co-authors, Mika Berger, who's currently in Brisbane at the moment, and Julia Perina, who's also here in Perth. I'd also like to acknowledge that we're meeting on Whadjuk Noongar land and pay my respects to their elders past and present. Pseudoscorpions are a cosmopolitan group of organisms. They're found in virtually every terrestrial habitat you can think of, except the polar regions. They occur from the intertidal zone up to the upper slopes of Mount Everest. This is a map showing type localities of all of the species described from around the world. And they're quite easy to find in many habitats if you know what you're looking for. And we're also aware that most of the families are seriously old. So the first pseudoscorpions have been recovered from Devonian shale sediments in New York about 380 million years ago. And they're quite recognizable as pseudoscorpions, even though they're placed in their own suborder now. And all of the Mesozoic fossils, those that are about 100 million years old, can easily be attributed to modern families. So it seems as though all of the families, or most of the families, had already evolved by the Mesozoic. This is a molecular phylogeny based on a transcriptomic analysis. We filtered out about 200 genes to produce the bulk of this phylogeny. There are a couple of tips here that have been added based on morphology where we didn't have transcriptomic data available. And I'm going to be talking on the family Hyaidae here today. All of those clades that are marked in with black circles here on the family terminals are those that have been recovered from Cretaceous ambers, particularly Burmese ambers so far. So we've got pretty good coverage of, at least in the fossil record. But the family I'm going to talk about today is a relatively small family. It's known as Hyaidae. There are only two genera. There's the genus Hyia, which occurs in Southeast Asia and in Sri Lanka. And there's also a genus called Indo-Hyia that was first described from the Western Ghats in India. It's also a species known from Madagascar, and they're also quite diverse in northwestern Australia, and that's what I'll be talking about for the rest of this talk. But just to recap on the genus Hyia and the age of the family, we've got the two described species from Southeast Asia and Sri Lanka, but we're also aware of an unnamed Cretaceous fossil from Burmese amber. This is an image of the specimen here, and apart from it being a little bit larger than the two species that are extant today throughout Asia, and a slightly different dentition of the teeth on the chela. That's the chela, the pedipalp that I'm pointing to there. It's virtually identical to the modern fauna. And this seems to be a recurring theme with pseudoscorpions, that we've got very sort of long-lived clades and very little morphological changes over the last 100 million years. So they're quite an interesting group when it comes to studying past patterns of biogeography as well. As I mentioned, there are species in India and Madagascar, which I won't refer to any longer, and concentrating on the Australian fauna. Specimens are rather rare in the genus Indo-Hyia. Most of them actually come from subterranean sampling, caves in the Cape Range area of Western Australia, but a lot of subterranean sampling has occurred due to environmental impact assessments, particularly in the Pilbara region of Western Australia, but also partly in some areas of Kimberley region in northern Western Australia. So we're kind of a little bit restricted, that it's not all that simple to go out and sample these animals. There are some epigean species that I'll talk about shortly, but most of them are subterranean. And as I said, rather rare in samples and sometimes of dubious quality. The quality, not in terms of the species themselves, but the quality of the samples, because every species of pseudoscorpion is beautiful, but you all know that. So we were able to generate a multi-gene phylogeny for the genus Indo-Hyia in Australia, and some outgroup specimens of Hyia that have been sampled from Asia. And there's a couple of things to note here. We've got 28 species represented on this tree. We've got 90 specimens. At the moment, we've only been able to analyse and run 3 different gene regions This is a RAXML analysis run by Julia Perina on behalf of this project. And as you can see here, we've got a couple of issues. First of all, the genus Hire is not sister to what we're calling Indo-Hire from Australia at the moment. But I think this is mostly due to the gene regions that we've covered so far, and also possibly the deep ages of these clays. Remember that Hire is Cretaceous at least. The genus itself is 100 million years old, and so that's going to make that Indo-Hire is even older again. We've also got a couple of issues down here there's a couple of samples, specimens down here for which we only have one gene region, and so I realised yesterday that perhaps taking those out might improve the topology of our tree a little bit. But that really doesn't affect the main thesis that we're planning to explain to you today. If we look at the distribution of the specimens across the landscape in the Pilbara and the Kimberley, we can see that there are certain clades that are cropping up all the time. And I should add that we don't have any sequence data for the Indo-Hires that are from the Western Ghats in India or from Madagascar as yet, so we're relying solely on the Australian component. You can see here there's I haven't got support values on this tree, but it's a pretty well supported clade here. We have a series of specimens from the Kimberley region of Western Australia, in fact all from one island. We have 3 species represented here in a clade. We have the epigean species of Indo-Hire from the Pilbara, of which there are 3 different species represented as a monophyletic clade. And that's sister to 3 species of troglodytic species from nearby Cape Range, which is a very interesting pattern that we weren't expecting to see. There's also a very big clade of Pilbara troglobites, subterranean fauna, this blue clade here. And it shows that they have not been primarily derived from the local epigean fauna in the Pilbara, and possibly, we haven't got a dated analysis as yet, but possibly indicating that this clade of subterranean organisms might be older than the epigean ones that are still up on the surface. As I mentioned before, we've got a couple of specimens down the bottom here with the asterisks that for which we have poor sequence data, so their placement is probably a little bit suspicious. And there's another Kimberley clade of epigean island-dwelling species at this part of the tree down here. So in summary, we can see that of this part of the analysis, we can see that the Kimberley is cropping up in different places in the phylogeny. Likewise, the Pilbara is as well. And we've got Cape Range, which is a relatively young landscape. It's only those caves, those limestones are only a few million years old. So they've probably moved in from surrounding areas to inhabit those cave systems. At the moment, we haven't been able to find any epigean representatives on Cape Range, not through want of looking, but they don't seem to be there at the moment. So I'm going to go into a little bit more detail about the 2 areas that we've been able to sample intensively so far. So the Kimberley region, in fact, it was the first representatives of the genus Indo-Hire that were ever collected in Australia back in rainforest surveys in the late 1980s through government agencies here in Western Australia. And there are a series of species, one of which appears to be moderately widespread, this yellow species here, that are mostly found in vine thickets, which are sort of dry rainforest habitats throughout the Kimberley. I would expect that once we go back and sample some of these older locations for sequence data, we might find the existence of additional species, cryptic species in this yellow population in particular, because we're seeing short-range endemism across the Pilbara in all the other species. At the moment, we've also got 5 different troglodytic species that are represented in our sampling from various places in the Kimberley. These include big cave systems to the northeast and to the south of the Kimberley. A lot of these we don't have sequence data for because the specimens date back to the 1980s, 1990s, and a lot of them are unsuitable for sequence data, or at least the methods, the Sanger methods that we've been using. But interestingly, on Irvine Island, in a series of samples from boreholes that are situated quite next to each other, we're able to find a transition of, this is a monophyletic clade here, we found a transition from fully eyed epigean species with relatively robust pedipalps through to a pair of quite blind troglodytic species with more attenuate pedipalps, all occurring in a local environment. Excuse me. So this seems to indicate that we've got a transition from fully eyed to troglodytic fauna occurring, as I said, right in the local landscape. Moving down to the Pilbara and the Cape Range, the most southern extent, we currently recognise 24 species, 2 of which have names, 22 are new. There are 19 troglobites and 3 epigean species. The epigean species, 2 of them are relatively widespread in the Pilbara. One is quite localised at the moment, but they've got eyes and relatively nondescript looking animals, and 21 different species of troglodytic taxa from across Cape Range and the Pilbara region. And looking at them in a little bit more detail, we can see that the scale here is, remember, is only 10 kilometres, and we have a complex of 3 closely related species here, separated on different ridges in this particular part of the Pilbara, which probably their isolation in these ridges over time has probably led to these speciation events. And the same thing occurs in other parts of the Pilbara as well. So we have extreme short-range endemism. They're restricted to individual geological landforms, the troglodytic species, and it's clear that there's been more than one incursion into subterranean habitats, especially in the Pilbara. So why study pseudoscorpions? Well, they occur in these geological formations that are being sampled, and through the environmental impact assessments, they're being sampled by regulation here in Western Australia. And this is what happens when mining approvals are given to mine areas. And basically, it appears that they're taking away most, if not all, of the available habitat for these individual species. So we're sort of racing the clock and trying to come up with conservation assessments at the same time while we're doing the systematics and the taxonomy. I'd like to thank the funding partners there, the collectors, including all the environmental companies who bring these samples back for us. It's been actually really fantastic working with them. Sequence data contributed by people who are listed here, my co-authors, and thank you very much for listening.
That was fantastic, Mark, as always. Has anyone got any questions? You can put your hand up in the chat or write it. I'll ask a quick question because we got a couple of minutes, Mark.
Sure.
With the things that you can't get molecular data from, has anyone tried doing combined analyses with the morphology? Do you have enough characters to do that? And has that been helpful in any way, maybe for dated analyses, things like that, or not really? Yeah,
good, really good question, Jeremy. Thank you. No, we haven't tried those analyses yet. Some of the taxonomic characters we use to separate these organisms are they're there, but they're a bit subtle. But codifying them is going to be a little bit difficult as well. And I should also add that some of the taxa that we propose to describe, we're going to be solely describing on sequence data because we have only got juvenile specimens, but they're so rare. The prospect of getting additional specimens from these sites, which are under a huge amount of pressure from mining activities, we think it's worthwhile going ahead with that.
Okay, thanks, Mark. We have a little bit of time for a quick question from Steve. Can you unmute, Steve? Or
great talk, Mark. Thanks very much. I'm interested in whether the subterranean species in the Pilbara region potentially evolved from other troglobites, or whether you think there were sort of more widespread epigean species that then became extinct for some reason.
Yeah, Steve, great question. We can't test that at the moment because we can't find any epigean species that are related to them. But in a paper that Kim Abrams published last year on schizomats in the Pilbara, she postulated an idea that the subterranean species have been derived from different epigean species that might have occurred in ravines and things since the Miocene, since it all started drying out. So we can't test it at the moment quite easily, but it's absolutely fascinating area to keep looking into.
Yeah, if you get a geographic pattern, I guess, to the situation, you know, the relatedness of species in the phylogeny, perhaps that indicates the dispersal along a valley or, yeah.
Yeah. I didn't go into details, but a lot of those clades are clustered in certain areas. So one particular part of the Pilbara will generate a monophyletic group.
Thanks, Mark.
All right.
Thanks, Steve.
Thanks so much, Mark. So we'll move on to the next talk.
Mark, there's a comment there for you from Andy.
I can't keep Andy awake for 10 minutes of the code. He gets bored after 1 minute and starts looking at his phone. So no, I won't. Thanks, Andy.